T-cell recognition of Mycobacterium tuberculosis peptides presented by HLA-E derived from infected human cells. The primary goal of this study was to characterize the primary protein structure of certain ligands in order to enrich our understanding of the relationship between MTB ligands and the cellular responses they elicit. Because HLA-E engages cell-mediated and humoral immunity through CD8 T-cell activation, the proteins were characterized based primarily on the CD8 T-cell responses generated post-Mycobacterium tuberculosis infection. HLA, or the human leukocyte antigen system, is the major histocompatibility complex protein expressed on the surface of human cells, which present self and foreign antigens and allow our immune system to distinguish self from non-self. A class of MHC found in humans called HLA-E presents pathogen-derived antigens to CD8 positive T cells, which become activated through their T cell receptor, resulting in T cell expansion and induction of immunological memory, characteristic of the adaptive immune system. CD8 T cells, which recognize and are activated by MTB antigens, do not respond exclusively on either classical or non-classical systems with varying degrees of polymorphisms. The response generated by non-classical and low polymorphic HLA-E induces reliable T cell activation, which can be elicited among a genetically diverse population. This property lends credence to the evolving concept of donor unrestricted T cells involved in vaccine therapy development, which do not rely on HLA compatibility. This is especially advantageous because under normal circumstances, the degree by which CD8 T cells response is elicited may be limited by donor-specific HLAs. Because vaccines exploit our natural immune system process, in order to develop immunological memory, a successful vaccine must take into consideration the mechanisms by which pathogens are naturally eradicated by our body. In similar studies characterizing MTB progression, T-cell-mediated immunity was identified as a major factor in controlling the clinical outcome of TB. Therefore, the primary goal of this study, which attempts to identify the specific ligands involved in activating T-cell-mediated immunity, is consistent with developing a non-traditional and effective vaccine strategy. Peripheral blood mononuclear cells were obtained by apheresis from healthy, latent MTB-infected and MTB-infected adult donors. Uninfected individuals were defined as healthy individuals with a negative tuberculin skin test. Individuals with latent TB were defined as healthy individuals with a positive skin test but no symptoms and no signs of active TB. And individuals with active TB were diagnosed and confirmed with positive sputum culture. This slide outlines the conditions for preparing the cultivation of MTB cells, specifically the H37RV strain of MTB expressing GFP. This slide outlines how monocyte-derived dendritic cells were prepared from PBMC donor cells. Cell line selection was accomplished by comparing how well each cell could present MTB-derived antigens to a human HLA-E restricted T cell compared to a dendritic cell. Soluble HLA-E producing MTB-infected U373 cells were electroporated and transfected with HLA-E103, where the transmembrane domains were replaced with mammalian-derived VLDL receptors. The stable transfectants were cloned and assayed utilizing ELISA via anti-beta-2 microglobulin antibody. The most suitable clone was grown in bioreactive conditions, and soluble HLA-E was collected before subsequent MTB inoculation. The GFP-expressing MTB-infected cells were quantified via fluorescent microscopy. The uninfected soluble HLA-E was purified using affinity chromatography and anti-VLDLR antibody. shLA-E were eluted from column using 0.2 m acetic acid. Ligands were eluted from the HLA complex via boiling at 75 degrees Celsius for 15 minutes, and the ligand pool was isolated from the alpha chain and beta-2 microglobulin with 3 kilodalton cutoff ultrafiltration. Characterization of ligand protein structures and lengths were accomplished utilizing LCMS and HPLC under conditions presented here. Liquid chromatography MS is an analytical chemistry technique that combines physical separation of chromatography with a mass spec. Ligand-bound soluble HLA-E molecules were identified through propagation of MTB-infected U373 cells and dendritic cells for analysis of peptide binding affinity utilizing MS. The parameters by which MS were used to identify ligands are outlined in this slide. The 28 identified HLA-E-bound peptides and their restricted T cells in which they elicited a response were further characterized by ELISpot assay of IF and gamma production. We can use this type of assay to quantitate the frequency of cytokine secretion for DC cells versus U373 cells. The parameters by which this was accomplished are outlined in this slide. The methods by which T cell lines were generated and harvested were derived from D481 PBMC are described in this slide. Figure 1 shows how selecting a suitable cell line for MTB ligand presentation was determined. In graph A, MTB ligand presentation to HLA-restricted T cells was compared between U373 and dendritic cells. Graph B demonstrates propagation of soluble HLA in milligrams in the aforementioned cell lines X days post-infection. Cell surface expression of pathogen-derived antigen on HLA-E depends on the suitability of leader peptide or binding affinity to soluble HLA-E. Binding affinity for a given ligand repertoire was characterized by abundance graphically represented in chart A. Abundance of leader peptide in TB-infected cells and uninfected cells was determined utilizing ion spectra where analyte intensity is plotted against mass to charge ratio. Graph B represents the length of HLE ligands in relative abundance and percentage, and Graph C represents peptide binding motifs generated by Sec2 logo, a web-based sequence logo generation method for constructing and visualizing amino acid binding motifs. These graphs demonstrate how MTB infection caused changes in the composition of the HLA-E ligand repertoire and increases MTB-specific ligands. The chromatogram and ion spectra developed in graph A and B were utilized to ascertain that the sequence presented in Table 2 were accurately deduced, specifically the sequence of the peptide derived from the highly conserved MTB protein RV0634A. Figure 5 demonstrates how presentation ability in the 28 identified MTB ligands compare in 4 healthy, 8 latent TBI, and 4 TB-infected donors. The graphs in Figure 6 represent the response elicited by RVO634A peptide in 10 out of 13 donors. The highest response was further analyzed to determine T cell restriction. Graph A shows donor T cell response to RV0634A. Graph B and Graph C1 illustrate degree of CD8 response to RV0364A elicited by cells stained with HLA-E tetramer. Graph C2 illustrates degree of CD8 response to RV0634A elicited by cells stained with HLA-B tetramer. Graph D1 illustrates negative peptide-specific staining of HLA-E-induced CD8 response to NKG2A in D481 cell lines. Graph D2 illustrates peptide-specific staining of HLA-E-induced CD8 response to NKG2A in PBMC cell lines and serves as a positive control. Graph E illustrates the IF and gamma response elicited by mismatched A549 cell line pulsed with the RVO634A19 peptide and used as antigen-presenting cell with D481 CD8 positive T cell lines. Table 1 outlines leader sequences associated with HLA-E. HLA-E presents leader peptides derived from other HLA class 1 molecules. These leader peptides comprise an optimized sequence that enhances antigen binding within the HLA-E group. This table outlines the defined MTB peptides associated with soluble HLA-E, which elicit a CD8 positive T cell response. Table 3 lists the response of IF and gamma ELISpot assay for each T cell response eliciting MTB ligand, which were tested in duplicate at 250,000 cells per well. The goal of the study was to identify and characterize unique pathogen-derived peptides, which upon HLA-E presentation activate HLA-E-restricted CD8 positive T cell response. The broader implication of reaching this goal was to determine a suitable vaccine target. This was achieved by utilizing a system which employs purification and collection of soluble HLA-E-bound ligands associated with MTB infection. In order to propagate HLA-E, a suitable cell line was selected based on how well each cell could present MTB derived antigen to class-restricted T cell clones, compared to primary human dendritic cells. The HLA-restricted T cell clone, D160, was able to make IFN gamma in response to MTB-infected U373 cells at levels similar to professional antigen-presenting cells such as DC. Likewise, U373 cells produce sufficient levels of soluble HLA-E molecules for downstream MS analysis. The primary structure and binding capacity of these collective ligands were characterized using MS. 28 Mycobacterium tuberculosis cellular signaling molecules derived from 13 different protein sources were identified. One in particular was highly conserved from the RV06348 protein and was recognized by 13 of the 16 donors in each category, including the healthy unexposed control group. HLE binding capacity can also be characterized by HLA leader sequence binding. This study also demonstrates that HLE can present pathogen-derived peptides of diverse lengths because of the cell surface expression of pathogen-derived antigen on HLA-E exhibits binding groove plasticity, which was provided by leader peptides. The 28 MTB ligands identified represented only 0.4% of the ligand repertoire during an MTB infection. The peptides identified and selected were consistent with the nature and the course of MTB infections, which utilize class 1 MHC loading pathways. In particular, half of the ligands identified were derived from a family of proteins called ESX, whose secretion system exposes endosomal MTB antigens to systolic conditions in class 1 loading pathways. This finding presented one limitation to suitable ligand identification, destined for antigen processing and ultimately presentation to class-restricted T cells. The identified ligands were further characterized based on the IFN gamma response. In 12 of the 28 ligands T cell activated responses, IFN gamma was elicited. This highlighted the ligand's ability to leverage a Th1 or cellular-mediated response. The remaining 16 ligands were not screened by enough donors to characterize T cell response. Ultimately, the study provided support for HLA-E as a plausible candidate for novel vaccine development. Here are my references. Thanks for listening.